Yeah, I just want to reiterate that I'm very happy to be interrupted with questions and all that, and just yell if I don't notice you. Let me stick this thing away. Okay, so Vivek introduced you to this. This is the central complex of the fly, a circuit that's responsible for navigation. I'm not going to take you all the way through it, but I want to particularly focus on connectomic related stuff. So Vivek talked to you about the discovery of the ring attractor in the compass system. That's had a huge impact. For example, here are the members of the theory center acting out the ring attractor in the fly. I think probably Vivek also told you about this very beautiful paper with Anne Hermanstadt, Jerry Rubin, and members of their labs. And I'm going to very much follow in line with this paper. They explored, what can you infer from the connectome of this structure about how it works? We know more now. I'm going to cheat like crazy. In other words, the reason I'm able to tell you what I'm going to tell you on the basis of the connectome is because I know the answer from other sources of information. But I think it's worth going back, even if you didn't get there this way, going back to the connectome and saying, what could I have learned if I was smart enough, looking at it the first time before that? Okay. So I want to introduce you to the heading vector, which I know Vivek talked about, just to get the pictures down of what's going on. So the idea is that a fly keeps track of its heading as it turns in the world. But when you look at it this way, it's kind of confusing because the heading is always, straight out the front of the fly. So what you have to keep in mind is what that means is that within this ring attractor, there's a representation of heading that actually turns in the fly's head. So on the left is the fly-centric viewpoint. There's actually a vector being rotated in there. And I will use these diagrams as I go along I just wanted to get the reference to that idea of these two pictures, the fly in the world, the fly in its own world. Okay, so we have a heading signal, and I think as Vivek also said, that gets passed down through the layers of this system or through the different components of this system. It goes to the protocerebral bridge. At that point, another set of neurons picks up the heading signal and through a transduction that I'm going to talk about in just a second, sends it to the fan-shaped body. The fan-shaped body also gets through these noduli that you can probably just barely see at the bottom, gets sensory information, and then it outputs a motor signal. So the idea is that particularly the fan-shaped body is a navigational computer, at least one of its functions, in which takes in sensory information, heading information, comes to a conclusion about what should be done, and sends out a motor signal to act and do that thing. Okay, that's a general framework. A cool part of this system is that it can all be parameterized by angles, and that's really what makes the connectome work so well for this system. So obviously, the ring attractor, the circle in the ellipsoid body can be parameterized by an angle that goes from 0 to 360, but as Vivek might have mentioned, you should really think of all of these structures. There are 4 of them, the left and the right sides of the protocerebral bridge and the fan-shaped body. Think of them all as circles. They're circles that have been broken open or annuluses that have been broken open. And so they all can be parameterized by angles that go from 0 to 360 degrees like that. And what the connectome allows you to do is align the angles as you go down and follow the signal through this system. Down, at least on my slide, on my thing. It isn't really down, but so that's what we're going to do. We're going to follow these signals through the system, and along the way, pick up what we can learn from connectomics. So the first lesson comes up with this first relay. This is something also that Vivek mentioned. I just want to do a little more math with what Vivek talked about. So the heading signal comes down. The EPG neurons that form the ring also project to the protocerebral bridge. And so you really should think of the bump that signals heading as 3 bumps. One that goes around the ring at the top, and then 2 on each side that go back and forth, and when they get to the end, they flip to the other side. So here, this blue dot would come off this end and flip over to here and go around this circle. Okay, so there's 3 copies of it, and as I said, it's those bottom blue dots that pick up the signal and then carry it down into the fan-shaped body, combine it with sensory information, and now we're going to start computing things that are relevant to the fly more than just its heading. And there's a relay set of neurons, this is one of them, that are called delta 7s that do a large part of conveying this signal from the EPGs, the ring neurons, the compass neurons, not the ring neurons, the compass neurons, down to these other neurons that project down. All of the signal doesn't go through them, but a large part does. Okay, and they have some interesting properties that I think Vivek mentioned. I just want to go into more detail. So what you can think of, as I said, because this system is all parameterized by angles, you can think of the signal coming in to one of these delta 7s being parameterized by the angle of whichever of the compass neurons happen to be projecting to that part of the dendrite. You can trace it back, and that's what I've called theta in, whatever angle that particular input neuron to the delta 7 goes. And likewise, you can think of an output angle. Where does this delta 7 deliver its output? The sort of white dots on that picture at the left, that's where it's giving its output. You look on these thing and you can assign an angle to the output of this delta 7. And so you can think of this transformation as taking in an angle and spitting out an angle. And think of it as a function. It happens to be a function, primarily at least, of the difference between those 2 angles. So this is like a transformation of the signal. To make it simple, I'm going to linearize the delta 7. And in that case, all you have to do to compute this function is to count the synapses on the input side and count the synapses on the output side and just figure out what that transformation is. In the fly, it's probably already been mentioned, the strengths of synapses are done usually by counting, or I should really say the strength of a connection is done by counting the numbers of synapses that contribute to that connection. And that's what's plotted here. So all the delta 7s have been plotted here as a function of the difference in the input and output angle of their thing. You count up the synapses and you get this curve, which is a cosine, quite accurately a cosine. So what that tells you, this isn't any old function. It's a constant plus a cosine of that angle. Okay, now you might say that's a curious fact, but actually I have to say in the fly, the fly work I'm saying is you should never say it's a curious fact. It always matters, and I think at least that this matters a lot, that this is a cosine. Let me explain why. Okay, so the idea is you start with an EPG signal. That's the compass neurons. That's the bump that signals heading. And you pass it through this, so it might look like that. I just made it a random shape and noisy. And you pass it through this filter. This is like a set of convolutional filters. And they're not just any old convolutional filters, they're cosine convolutional filters. So you pass it through that, and then you integrate over the whole ellipsoid body ring, right, over the whole circle, because those are all the possible inputs that can come in. And that'll give you the output at a certain angle that comes out of this delta 7. And due to a property of cosine convolutions, that will become a cosine, right? It has to be a cosine. It doesn't matter what you stick in, out will come a cosine. And so these delta 7s seem specifically built to take any old signal you want and turn it into a cosine. Yeah.
Just to be absolutely clear, so the theta out is an intrinsic property of each one of those bridge cells. Each one has its own theta out.
Exactly.
And then you're computing the output of that
you'll
cell.
You'll see when I show you, these purple lines have an angle assigned to them. Yeah. Okay, so now, this is a kind of a classic example of connectomics. This is suggestive, right? This is simplified. I linearized the delta 7. I'm ignoring other possible sources of input. So you got to check it. But in fact, if you do check it with physiology, it's actually true. So these are the neurons that are the purple lines. So what the angle, these different colors are different members of this class of neurons. This is, one type of PFN. And those are the angles that I called theta out. Okay, and so if you record the activity of these neurons, let's say in calcium or something, you really do see a sine wave, sinusoidal shape. It can be of different phase and different amplitude, but that's what you get. So this was real. And what does it mean? I think Vivek probably mentioned this, but this idea had been floating around for quite some time. You could have an array of neurons distributed, in this case, across space. That's at the bottom of this diagram. Their activities could be up and down in the shape of a sinusoidal pattern, like you see for these PFN neurons. They're going to have a phase of that, and they're going to have an amplitude. And the idea is that this maps one-to-one onto the length and the angle of a two-dimensional vector. So we're going to talk about navigation in this plane and a 2D navigation. And that's called a phasor representation. So that's the key to this system. Now, you can ask me later about up and down, right? Flies, after all, fly. And this system does not do up and down. And I don't at least know, I don't think any of us know where the up and down system is. This is lateral, if you want, navigation. Okay, so this thing, please remember this, because this is the key to understanding everything, that vectors are represented as phasors.
So, you bring in length here. Is that something that's modulated in the...
Yeah, you'll see, it's coming up, yep. Okay. All right, so let me start off with a representation of sensory input. So these guys here, maybe I should go back for a second. You'll see this in a second, but I want to introduce it. These cells are the ones that carry the heading information. It comes in here. They carry it down into the fan-shaped body where they deliver the information, but they also combine it with sensory information. And all this stuff down here is at least one of the places where they get sensory information. Okay, so I'm not really going to talk too much about this study, but I'm going to use wind as an example. And the stuff I'm talking about from now on, I think, until the very end, is done with Gabby Mayman and members of his lab that I'll point out along the way. So a fly, this is a fly out in the breeze. There's a wind blowing straight in its face, and flies detect the wind through their antennae, through this a torsion on their antennae. So in this case, the fly's two antennae would be equally disturbed by the wind, and they would report that the wind is straight out in front. And in fact, the antennae project the wind onto 45-degree axes. So you can think of as a 45 reference frame here, and the antennae are reporting projections of the wind velocity onto those two things. So there's been a lot of beautiful work, by the way, on this system by Kathy Nagel and members of her lab. Okay, so now if the fly turns, of course, now the wind is going to affect the two antennae differently. And the question is, can the fly know that the wind didn't actually change, right? The fly turned, the wind didn't change. So the fly, in fact, does construct a representation of the wind that it doesn't change in its brain, that in a case like this, when the wind doesn't change, and can detect the difference between turning, causing the wind change, and actually the wind really did change. And to do that, what you have to do, obviously, the input, the sensory input is changing. That's because the sensory input is in egocentric coordinates, and it changes with the rotation of the body. What the fly has to do is build an internal coordinate system on which to project that sensory data, and it has to make sure that that system rotates when the fly rotates. Now, the blue vector here is the heading vector that I already showed you, that is in the ellipsoid body. So the fly has to construct a coordinate system that rotates along with its heading vector. And what I want to do is show you how that happens and how the connectome helps us to figure out how it happens. And then what has to happen, this is Srini's question, I'll show you that briefly, is that then these projections have to affect the lengths of those vectors, right? And then you've got a representation of the wind now in allocentric coordinates. So for example, in the example I gave you, nothing would change. There would be a signal that the wind's still in the same direction it always was. Okay, so this was actually worked out initially not for the wind, but for optic flow. Optic flow, though, is very similar. When a fly moves through the world, the optic flow that it sees is projected by the eyes onto 45-degree axes, and it's exactly the same story, right? If the fly turns, it drifts, let's say, in the optic flow, it's the same idea. Okay, so this is work of Chung Liu. Also, and Rachel Wilson lab also worked on this. Okay, so how do the PFNs do that? Yeah.
Sorry, briefly.
Yeah.
The difference between the wind and the optic input, right? For the optic input, you have the flow field, but for the wind, it could be the external wind or the wind that the animal induces by flying itself, like it would be for the optics, right?
Yeah.
So it should be, I would expect there is a difference between the two, because one you have to subtract self-motion, and the other one
Yeah, so this is just, think of this as just direction. In the velocity, certainly you would want to do this, but ultimately, this is sort of related to Srini's question. In the end, ultimately, this is going to be a direction signal. And so in optic flow, basically, in the optic flow case, it's sort of, in order to detect like drift, you know, if you're being blown sideways by a wind, something like that. So it ends up being the same. Okay, so here I mentioned PFN neurons. There are different types of this. The wind is one type, the optic flow is another type. I'm just generically going to do this for sensory input and not go into subtypes of these PFN neurons. So the idea, I already mentioned this, is they get a heading signal up there. They get sensory information down there. And the sensory information is in egocentric coordinates, but they're going to use the knowledge of heading that they get to turn this into this egocentric output and report the egocentric direction of whatever the sensory information is. Okay, so I'm going to give you 3 pictures of this system, and we'll see how it works. And yeah, I got plenty of time. Okay, so this is obviously the anatomical picture. These are all pictures actually from this paper by Vivek and the group, the Big EM paper. And here is a sort of a physiological picture, except it's just drawn. It's a schematic, but it's true. And then I'll show you the third picture in a second. So the idea, I already said this, is you have a bump in the ellipsoid body. That's the heading signal. That is conveyed to these PFNs and results in a sinusoidal shape. It's a little truncated there, but sinusoidal shape within the activity across these regions. And they're yoked together. So if you image this and the fly turned, what would happen is the bump would turn and these guys would slide over by equivalent angle and they would slide back and forth in concert with the bump. All right, so now remember, the idea here is to translate sinusoids into vectors. And that means that each of those sinusoids represents a vector. It's parallel to the heading vector, and they all rotate together. So now if the fly turns, you know, they all go together. So it seems like, you know, we already had 3 copies of the heading, and now we've got many more because there are a number of different types of PFNs. So what are we doing? Copying this over and over again. And here is where the solution comes in the connectome. This was realized in the big connectome paper. I think Vivek mentioned it. And that is that if you follow this signal, it's received up here, but the business, the computations are done down there. If you follow this signal, what you find is a shift. So here, rather than showing in those other diagrams, I was showing you all of the PFNs, and they are, I should, you know, just point that out. Maybe I didn't really say it. They're called columnar neurons, right? So there's one here, goes here, purple here, you know, blue is here, right? They're all lined in columns, and that's the whole set. But here's just one of them. Sorry, we've got to go through all this over again. Here's one of them. And you can see, right, this is where it picks up the heading signal, and it picks it up. This is the theta out, right? That this particular PFN picks up its heading signal at a particular theta out given by wherever it is in the protocerebral bridge. And then it gets sensory input here. And in fact, this is the case of the optic flow. This purple neuron is a neuron that carries optic flow visual information. This green one, sorry, it's not purple, green one. Okay, and then here's where it puts its output out. And that output is going to go into representing the sensory information that whatever this is. Okay, now, if because of these angles, right, this is like 0 to 360, and this is 0 to 360, you can align this position and angle with the same angle down here. And if you do that, I think as Vivek told you, you would conclude that it missed its target. It didn't go to the analogous column. Instead, it went over by an amount that's 45 degrees. So there's a 45-degree shift here. And if you looked at one of these on the other side, it would also be 45 degrees shifted, but the other way. So in other words, if I go back to my triple view, if you follow this signal down, rather than two sine waves that are identical, you get two sine waves that are shifted by 90 degrees, 145 one way and 145 the other way. All right, so what's going on? Well, what's going on is obvious if you think about this phasor idea, right? Shifting the phase of a sine wave is equivalent to rotating a vector. So now if I give you this third column, what's happening is you're rotating two vectors by 45 degrees, and you've got yourself an orthogonal coordinate system, and that orthogonal coordinate system is tied to the heading. So that's exactly the system. I'm going to make the vectors a little longer, but that's exactly the coordinate system that we needed, right? It rotates when the fly turns, and so the system would look like this when the fly turns. Okay, so then all we have to do to finish the job off is to make sure that when sensory information comes in, this is what Srini was mentioning, that these guys have got to lengthen by appropriate amounts to represent the projections of the wind or the optic flow, whatever it is, onto those axes. And that's exactly what happens. So Chung, for example, measured this. Its cells measured it. What you see is when you put in sensory information that's skewed, one of these sine waves across the fan-shaped body gets big, one of them gets smaller. We know what's the translation of that into vector language. That means that the vectors get either smaller or bigger, and you can compute how much and find that, in fact, if you sum those vectors, you get the allocentric direction. I'll show you that in a second. So that's how the system works. Yeah, okay. And let me just say that, how do you add the vectors if you haven't done any phasors lately? If you want to add vectors and you have a phasor representation, so here's the red vector has been represented as a phasor, the blue vector is represented as another phasor. If you want to add the vectors, all you got to do is add the phasors. So that means you just have to take the responses of these cells and add them up in a target cell, and you're going to get a representation of that red vector on the previous page. So that is done for optic flow, at least, within a set of neurons called H delta B. They receive input from these PFNs that I've been talking about. They develop a hotspot, a bump-like of activity. But that one, it doesn't represent heading. It represents the allocentric direction of optic flow. And here's just an example that it works, right? So this is just measuring the location of the bump in H delta B, and then those green things are taking the PFN signal and adding it up vectorially and all that. And you can see it agrees very well. Okay, so that's how, good. That's how the input comes in. Yeah.
As far as the implementation, like you showed the two different signs that were induced by the purple arrows change their amplitudes differently. But that's sensory information that's leading to that.
Mm-hmm.
So it's not that the output coming down changes amplitude.
Yeah, exactly.
It's
Yeah.
the sensory combines with it.
Yeah, yeah. So these guys and these signals have an effective multiplicative interaction. And there are all sorts of tricks to pull that off, which I won't talk about, but I will in a second. Okay. All right, so just let me figure, yeah, where I am. Okay. All right, so I want to get on to this motor output idea, right? Obviously, you have, let's say the fly just wants to go upwind. That's a simple one. So we might have a hot spot of activity here that represents the wind direction, and the fly wants to go upwind. It better emit a motor command saying, turn in the direction of the wind. And then, in fact, that's exactly what I'm going to describe. Okay, but I want to talk about another thing, because if you think about it for a bit, you would say, okay, it's very cool, you're representing vectors by cosines, but you're passing them through, in particular in here, through all sorts of different neurons and nonlinearities and all that. Isn't that cosine going to get mutilated along the way? So, as it passes through here, it just doesn't seem like it's not going to pick up harmonics and get phase shifts and all sorts of horrible things that are going to screw this up. And so this part of the talk is to give you an idea of the tricks that the system uses to avoid that from happening. And it could have tried to do that by very carefully tuning all the nonlinearities so the bad terms cancel. It would be a very hard problem. That is not what happens. It does it through the connections, through symmetries of the connections, and you'll see that in a second. Okay, so what we want to do to get a motor output is we need a goal, right? The fly has its heading signal, but if we want to look at turning towards a goal, we have to have the goal, and this is. So this is a representation of what the heading signal looks like, right? When the fly turns, it turns inside the head of the fly. But this is work done, again, with Gabby Mayman and Peter Muscles-Pires. Peter first found a goal signal in another set of neurons within the fan-shaped body. And that goal signal can be distinguished, there are a bunch of tests that I'm not going to go through, but what distinguishes it is that the fly turns, it doesn't turn, at least if the fly maintains its goal. So in other words, if I wanted to walk over here and I got turned, my goal wouldn't change, my heading would just change. And that's what happens with this signal. So we have a goal signal, and now what I really want to go through with you is, given the goal signal, if the fly does get blown off course, it has to compute it has to compare these two directions and realize I need to turn to the left to get back on course and emit a command that generates a left turn. Okay, and this was also a subject of a companion paper by Rachel Wilson's lab. All right, so this is done by another set of neurons called PFL3 in this particular case, that look kind of the same with an important difference. So these neurons pick up the heading signal again in the protocerebral bridge. The heading signal is critical for all this, right? Because it's the reference for everything. It picks up the goal signal within the fan-shaped body. Remember, the PFN neurons put their output there. In this case, it's getting an input, the goal signal, and puts its output out in a structure called the lateral accessory lobe, where it connects to descending neurons that generate things like forward motion, turns, things like that. So this is a kind of a motor output there. Okay. There are 12 of these guys on the right side, 12 of them on the left side. Because Vivek, you did it, you made everything from the front of the fly. Where is Vivek? Right? And we do everything from the back of the fly, thereby ruining my life. But anyway, this is the right. So 12 right and 12 left, and basically the idea is that the motor output is the sum of all them, and that the turning signal then is a difference. And again, this was all inferred in Vivek's paper. In fact, it's been inferred for a long time, starting with the Stone and Webb paper, a remarkable paper that, again, just kind of looked at the anatomy. It wasn't even a connectome, looked at the anatomy of this thing and kind of inferred the answer and got it right. Okay, so how does the system work? I got to remember what I'm doing. Oh, okay. I'm just going to do I've been, ignoring the experiments, which is unfortunate. But let me just tell you here how this is done, because the key in doing any goal-related thing is you got to know what the goal of the fly is, and the goal is in the head of the fly. But flies have a behavior where if they're overheated, they'll just pick a random direction and walk in that direction for a long time. And that's what you're seeing here. In fact, these are the traces from the recordings that Peter used for this. And so I can see, I think you can see that you can infer a goal direction from these just by looking at which way the fly went. So it's a fortunate behavior. And the first thing he did was just image the summed activity on the left and the right sides. Those should be kind of the turning signals. And here's what he got. He got a very sinusoidal-looking curve, another sinusoid. And this is a sinusoid very deep into the system, by the way. It's a sinusoid as a function of the sort of heading error, the difference between the heading and the goal. So this is what's going to generate a turning signal. And in fact, if you just take the difference of those, you get a really nice sinusoidal turning signal. It looks like this. So it's basically a sign of the goal minus the heading. So it says turn right if you're on one side, turn left if you're on the other side. If you're right on target, don't do anything. These curves are a little funky back here because the flies don't very often go 180 degrees from their goal. So there's just not very much data down there. Okay, so how did this happen? How did you get a sinusoid here? I got lots of time. Okay, so Peter recorded also individually from these cells, intracellularly, a much more difficult thing to do. And their activity is going to depend on heading and goal, right? Those are their inputs. They get a heading angle here, they get a goal angle here, so it's going to be a function of that. And he looked at that function. So let's say, it's cell I, just to give a label, and the function looked like this. The response of this cell, the spiking of this cell as a function of goal, was maximal at some preferred goal direction. So different cells have different goal directions. So, one cell might have its maximum for a goal direction there, another cell for there. But it was a function of that. And in addition, it had a very similar thing in the heading. So here this HRR is also the preferred heading angle. So when the fly was at a particular heading for that cell, you got the maximal response. And tailed off from that angle. The reason that I labeled it with an R rather than preferred is because if you do the cells on the left side, you get another set of angles. So they're not the same. Okay, so this occurs for 12 cells on each side, right? And Peter could not record from all of them or identify all of them. And so in order to get these angles, which we need to finish the story, we had to do connectomics. So that's where connectomics is going to come in here. But once we have these functions, the idea is you just sum them on each side, and you should have the answer. So in other words, these data should be explained by taking those functions and adding them over the 12 cells, and likewise for the left and the right, adding them over the 12 cells. And the key is, though, sorry, I thought I had it labeled. The key is to know these angles, right? And what we're going to do, so in reality, what we did here, and maybe if I have time, I'll show it to you, is actually measure this function, determine these angles from the connectomes, and just add it up. But what I want to do is show you how you can sort of finesse that, without even knowing the function, determine the answer. And the reason, I think that's important is because you might argue, if I showed you the other way, you might argue, well, that's the magic function, that, again, combines all this stuff in exactly the right way Turns out it doesn't really matter what this function is. As long as it's a nonlinear, relatively smooth function, the system's going to work. And work means that you get these curves and you get a sinusoidal turning curve. Okay, so let's go through the argument. Whatever this sum is, it's definitely a function of two angles, the heading angle and the goal angle. And because of that, it can be written as a double Fourier series, right? This is just, standard Fourier series for a function of two angles. All right? And, I'm going to make the argument that these functions are relatively smooth functions. They get inputs that are more or less cosine shaped. And so in this sum, there are not going to be high harmonics. So in fact, I've listed all the lowest harmonics. And I'm going to assume that this dot, dot, dot is not too big, okay? All right, so that's what you would get. And the key is that, what we see in these responses is just those three terms, right? So why? How did the system get rid of all these other terms? And again, you could say, well, it just added everything up properly. Well, it kind of is the answer. But you'll see it's all in the connectome, the fact that it doesn't. And I think you all could guess an answer here, which is that these terms are special because they're rotationally invariant, right? If you change G and H by the same angle, they don't change, whereas all the other terms do change. So if I was building this model, or if you were building this model, what you would say is, I know how to do this. All you have to do is make sure that these angles here are as rotationally invariant as possible. In other words, put them uniformly around the circle, as many as you've got, and this is going to work, right? Especially if you don't have to worry about high harmonics. And so, in fact, that kind of looks pretty good, because what you do is you now have to determine these angles, and let me just point out how to do it. Basically, to determine these angles, you've got to trace from the goal cells that found here up to these PFL3s and see how they align in the columns. And you have got to trace the heading signal all the way down from the compass neurons all the way down here to try to figure out which heading up there is going to make this particular cell fire the most. But that can all be done by just tracing through the connectome. And if you do it for the goal cells, in fact, it looks like, it's right. The goal cells are just evenly distributed every 30 degrees. So they have this symmetry, where the parentheses here just means, modulo 12. Okay, so when I saw this, I immediately built a model saying, well, look, we got it, right? The H's, the preferred headings, are also going to be uniformly spread around. And then it's going to work, because we don't have an exact angular translation invariance, but we got it pretty close to it. So, yeah.
So the way you got those numbers there, the angles, is based on the connectome weights, the synapse counts?
Yeah.
Propagating?
Yeah. Just see who's connected to who.
But
and it's all within columns.
And the weighting, right? The cosine sort of tuning and so the precise details of exactly, what synapse counts showed up where.
Yeah.
If we sort of propagate those weights forward, should give us like precise angles.
So okay, these are rounded off. The angles that I'll give you are either sort of center of mass angles of a distribution, because the connections aren't perfect, or max. And then they're quantized in columns.
Yeah, I see the caricature that you're building here. I'm just wondering if from the weights there's a more precise sort of estimate of the preferred angles that can be made.
Yeah. So there's a sort of a question, what do you do for the slop on the sides? I guess, it comes down, would you do a center of mass, in which case you wouldn't get even numbers, or do you do max?
Well, I'm just curious what it comes out to.
Yeah.
And how much variability there is and what the sort of...
Yeah, so few degrees. I mean, I'd have to look up. I don't remember the numbers.
Yes.
Certainly.
And how carefully, they're matched on the
Yeah.
left and right side.
Yeah, yeah.
Just,
So let me come back to that, actually, because I make a comment about that. Okay. So I built a model in which these guys were also just evenly spaced around the circle. I was so proud of myself. In the old days, I would have published that model and been even more proud of myself. But Peter just immediately said, look at the connectome. You're wrong. It's not the way it works, which was very perturbing. So to look at the connectome, just give you an idea. I mean, I guess you had beautiful, even more beautiful pictures. But the idea is, these are some PFL3 connections. So remember, what we got to figure out is the signal goes from the compass neurons to these delta 7s to these guys. And the tricky part was figuring out how these guys connect up with the delta 7. So there's one of the delta 7s, there's another delta 7, and there is, let me just see where you are. Yeah, this is the irregular one. Okay, so I'll just mention, this one drove us crazy because these guys, the protocerebral bridge has glomeruli, and this is one, and this is one. And this guy goes right between two glomeruli, which it shouldn't do, and it caused confusion with us, also with the Wilson group. But it turns out there's also a delta 7 that goes right between the two thing. And I was asking this morning whether in the flywire it also has this weird feature. Okay, anyway, you do this, you get a matrix. Again, this is the max matrix. And you get these angles. And if you first look at these, you just say, oh my God, what is this thing doing? There's a number, then there's a repeat, then there's a number, now there's a repeat. But it has a symmetry. Fly is not so dumb. It has this symmetry. If you shift by 3, you get a 90-degree shift. And it's a repeating pattern. And so it's a kind of a weird way to get this rotational symmetry, but it does. And the idea is that since you're summing over I, you can shift all you want. It's going to be a symmetry. So this is going to be a symmetry of this system. If you look at the ones on the other side, they also have this symmetry. And between the two of these guys, they also have a funny relationship that one of them is the negative of the other one going backwards. Okay? So these symmetries are going to save the day. This, and this. Because, let me see, I think I have it here, because they imply that this function has a 90-degree symmetry. If you shift the heading and the goal by 90 degrees, you're right back where you started. That's not a perfect, rotational invariance, but it's good enough to get rid of all those terms. They don't obey it. Okay. So the idea is that this system it relies on symmetries. You say, how did these cosines get preserved? It's not because there's magic nonlinearities that don't distort cosines. It's because when you sum things up in the end, and because of the symmetries within the connectome, you can't lose. And by the way, this also gets rid of a lot of these terms, these ... terms. So in fact, when we do this, these things turn out to be sinusoidal within a percent or two. Okay, so where am I? Okay, so the idea is that from this argument, if you were smart enough, you could have just looked at the connectome and say, this has to be fit by a function like this. Because of this symmetry, this has to be the flip of it, which if you notice it is, right? This is just the other one flipped. And that tells you that when you take the difference, you can only get the antisymmetric part, so you have to get a sign. And again, that's extremely accurately true in the data. Okay, so let me sum up, and either we can have a discussion, or if you don't have questions, I'll do a little bit more. But I want to kind of give you an overview of this system, how I think we all view it, and what remains in a way. So what I've shown you is that sensory information can come in, combined with heading information, and result in a hotspot of activity, a bump, that reflects some kind of sensory relevant thing like wind direction, optic flow, things like that. And that involves an ego to allocentric coordinate transformation. This is an egocentric coordinates. This gives the fly the position in the world, and the result, it gets this allocentric. This kind of made me think more about, I'll mention one thing. If you just take the hemibrain connectome and you count synapses, and say, where are all the synapses going? Where are the majority of synapses? You get things that you would all guess, vision and olfaction. But number 3 on the list is this thing, is input to the compass. And so I think it really points out a kind of a sense that we all have that we completely take for granted. And that is the sense of where am I in this room? You know, the sense I know where I am, I know when I rotate, I know where all you are, which I like to call alloception, which is not a word. And you know, there are human conditions like hemi-neglect where people lose this sense, at least on one side due to a stroke. So keep in mind that you have this sense of where you are in the world, and it's incredibly important. It's incredibly important to the fly, and if you silence the heading signal, there are lots of things a fly can't do, which are related to navigation. Okay, so that's that transformation. Now, there are multiple sensory inputs here. There are multiple kinds of PFNs, so there are multiple things represented. And you know, I think of these as various things that would be relevant for navigation, including perhaps memories. Then there's a goal direction signal, and I talked about how that gets out and generates turning. What remains, I think, and I should mention, that undoes the transformation, right? If I know that you guys are out there, if I want to turn, I have to go back and say, well, except I'm facing this way, I better turn left. So you have to undo the transformation. So what's left is, you know, how do you get from that general grab bag of different kinds of places that could be the fly's goal to the actual goal? And here, I think Vivek mentioned these guys. There are these tangential neurons that I think we all think of as control elements that could select one or the other of these guys and amplify it and make it the goal. And that actually solves a kind of a riddle here. And it's related to Srini's question at the beginning. So I showed how when the sensory information and the heading information get combined, it distorts the size of the amplitude of the sine waves in a very sensible way. But when you get to this representation, it's hard to see the amplitude of the sine being related to actual sensory information. And this is what I was saying to you, Suzanne, that for example, if you increase the wind velocity or increase the optic flow, this red dot isn't going to get any brighter. And I think that has to do with the fact that at this point, you want to use amplitude for a different purpose. You might want to boost the amplitude of this signal so that it affects the goal more strongly. So I think there's a flip from sort of true vector calculation to kind of unit vector calculation with weights on the unit vectors rather than sensible amplitudes, meaning geometric structure within this thing. Okay, I think I will just stop there. There's something I promised you I would say. Yes, I'll just end with answering Srini's question. So flies are not fantastic navigators, but other insects, I think Vivek showed you the ants, are fantastic navigators. And I would predict that this system is not that different for those animals, but it's wired much better. In other words, where you see slop, for example, in those angles that I showed, I would bet in the desert ant, you won't see any slop. It'll be perfect. Okay, I'm happy to have a discussion, whatever. Stop there.